A growing trend in modern pet care involves owners feeding their pets plant-based diets to match their own ethical choices. While dogs and humans can adapt to this, applying this diet to a domestic cat is a fatal metabolic error. We classify cats as obligate carnivores, a term that represents a rigid, inescapable metabolic limitation. This entire limitation hinges on a single, indispensable amino acid, taurine. Most mammals possess the internal machinery to synthesize this crucial compound from other building blocks. The domestic cat, however, cannot produce enough of it to sustain its own life. Without a steady external supply, the cat's physiology rapidly deteriorates. The immediate clinical consequences are severe, presenting directly as fatal heart conditions and irreversible ocular diseases. Feline nutrition is governed by uncompromising enzymatic laws. Any deviation from an animal-based diet directly induces a systemic physiological breakdown. To understand why this deficiency is lethal, we have to isolate the three biological systems in a cat that demand extraordinary amounts of taurine. The first is the myocardium, or heart muscle. Here, taurine regulates the flow of calcium ions, providing the exact mechanism that allows the cardiac tissue to contract and pump blood effectively. The second is the retina. Taurine maintains the structural integrity of the photoreceptor cells, ensuring they can absorb light and transmit visual signals to the brain. The third is the liver. The hepatic system relies heavily on taurine to conjugate bile acids, a process strictly required to digest and absorb dietary fats. Because the feline body cannot synthesize new molecules to replace what is used, these three organs constantly drain the cat's limited systemic reserves. These specific tissues are the first to undergo catastrophic failure. Their massive, continuous metabolic demand simply outpaces a non-existent internal supply. To see exactly where the cat's metabolism fails, we first need to look at how this biochemical pathway operates in omnivores and other carnivores, like dogs and humans. In a standard mammalian system, the process begins with cysteine, a foundational amino acid easily acquired from general dietary proteins. This biochemical flowchart illustrates the first enzymatic step, where ingested cysteine is oxidized into an intermediate compound known as cysteine sulfonate. Then comes the critical second step. An enzyme called cysteine sulfonic acid decarboxylase, or CSAD, decarboxylates the compound, driving the chemical reaction forward. This leads directly into the final stages, where the resulting hypotaurine is oxidized to create the finished product, taurine. This highly efficient internal assembly line acts as an evolutionary safety net for most mammals. It ensures a steady baseline supply of taurine even if their daily diet lacks it. When we examine the feline model, this reliable baseline collapses entirely. By tracing this same biochemical flowchart for a cat, we can locate the exact point of genetic failure. The breakdown occurs right at that crucial second step, with the enzyme cysteine sulfonic acid decarboxylase. In the feline liver, this specific enzyme has extremely low, almost negligible activity compared to other species. Because of this severe bottleneck, the accumulating precursor cannot proceed. Instead, the cat's metabolism diverts the chemical flow into a competing pathway, turning it into pyruvate. This specific genetic bottleneck permanently disables the cat's ability to be metabolically independent. The animal is forced to outsource its biochemistry to its prey. In the wild, the feline species compensates for this broken internal pathway through highly specialized hunting. To survive, the cat must ingest taurine directly in its fully formed, bioavailable state. Raw animal tissue, specifically red meat, hearts, and other organs, is the only sufficient natural source of pre-synthesized taurine. Plant-based proteins are biologically useless to the cat in this regard. Even if a vegan diet is artificially loaded with the precursor amino acid cysteine, the CSAD bottleneck ensures the cat can never convert it into the taurine it desperately needs. A cat's status as a predator is a strict biochemical mandate dictated by an absolute need for exogenous animal tissue. When this strict dietary requirement is ignored, the biochemical bottleneck directly manifests as macroscopic tissue damage. Without taurine to regulate calcium in the heart muscle, the feline patient rapidly develops dilated cardiomyopathy. A healthy heart contracts tightly and efficiently. In contrast, the taurine-depleted heart becomes dilated, its muscular walls stretch thin, and it struggles to pump blood throughout the body. Simultaneously, the deficiency attacks the ocular system, leading to feline central retinal degeneration. The photoreceptor cells undergo irreversible structural degradation. Hyperreflective lesions form across the tissue, leading directly to permanent blindness. Taurine deficiency induces a highly targeted, catastrophic failure of the animal's most vital organs. Feline dietary needs cannot be negotiated, and they certainly cannot be substituted with plant-based alternatives. The inability to synthesize taurine is a hardwired, unchangeable genetic trait. This is the true clinical definition of an obligate carnivore. To treat a feline patient successfully, a clinician must fully understand and respect these rigid biochemical laws.